Today I'll be talking about cytoskeleton. As per the syllabus, we have the type, structure and functions of cytoskeleton, the accessory proteins of microfilaments and microtubules. Now, first let us know what is the cytoskeleton. All cells have to be able to rearrange their internal components as they grow, divide, and adapt to changing circumstances. Eukaryotic cells have developed all these spatial and mechanical functions to a very high degree, and they depend on a remarkable system of filaments called the cytoskeleton. Now here we begin by describing the three main types of filaments, the basic principles underlying their assembly and disassembly, and their individual peculiarities. Most animal cells have three types of cytoskeletal filaments that are responsible for the cell's spatial organization and mechanical properties. Intermediate filaments provide mechanical strength. Microtubules determine the positions of membrane enclosed organelles and direct intracellular transport. Actin filaments determine the shape of the cell surface and are necessary for whole cell locomotion. But these cytoskeletal filaments would be ineffective without the hundreds of accessory proteins that link the filaments to other cell components as well as to each other. This large set of accessory proteins is essential for controlled assembly of the cytoskeletal filaments in particular locations, and it includes the motor proteins, remarkable molecular machines that convert the energy of ATP hydrolysis into mechanical force that can either move organelles along the filaments or move the filaments themselves. Intermediate filaments. Intermediate filaments are rope-like cytoplasmic filaments of about just 10 nanometer diameter. These filaments are found in many metazoans, including vertebrates, nematodes and mollusks, but not in plants and fungi. Unlike the actin and tubulin proteins, the intermediate filament proteins are chemically heterogeneous and show species-specific variations in molecular weight. The principal functions of intermediate filaments are structural, to reinforce cells and to organize cells into tissues. Unlike microfilaments and microtubules, intermediate filaments do not participate in cell motility. All intermediate filaments share a common structural organization. The individual polypeptide of intermediate filaments is an elongated molecule consisting of a non-alpha helical N-terminal head domain, a central alpha helical rod domain, and the non-alpha helical C-terminal tail domain. The central rod domain consists of long tandem repeats of a distinctive 7 amino acid sequence called the heptad repeat. Polypeptide chain forms a parallel coiled-coil dimeric structure with another. Two dimers then line up side by side to form an antiparallel tetramer of 4 polypeptide chains. Tetramer, the soluble subunit of intermediate filament, further organizes to form higher level organization. Tetramer is analogous to the alpha-beta tubulin heterodimer of the G-actin. Unlike the actin or tubulin subunits, the intermediate filament subunits do not contain a binding site for a nucleoside triphosphate. The antiparallel arrangement of dimers implies that the tetramer and hence the intermediate filament that it forms is a non-polarized structure. So to sum up, monomer, which is a single polypeptide chain, they form the coiled coil dimer, which is hetero or homodimer, and parallel coiled structure. Now all these form the tetramer, that is the staggered arrangement of dimer and lacks structural polarity, and all these things together form the intermediate filament, which is about 10 nanometer in diameter. Now look at the types of the intermediate filaments. These are the types of intermediate filaments that is encountered. You can find it's under the heading nuclear, then vimentin-like, then epithelial, axonal, and here I have discussed about the component polypeptides and the features and the locations of each and every type. Now I will be talking about the microtubules. This is a type of the cytoskeleton. What are microtubules? Microtubules are long, hollow cylinders made of the protein tubulin. With an outer diameter of 25 nanometer, they are much more rigid than actin filaments. Microtubules are long and straight and typically have one end attached to a single microtubule organizing center, MTOC, called a centrosome. Microtubules are actually hollow cylindrical structure, approximately 25 nanometer in diameter, as I have said. Now they are present in all eukaryotic cells and play crucial roles in the determination of cell shape and cellular motility. The basic building block of the microtubule is alpha beta tubulin heterodimer, composed of two 50 kilodalton subunits known as alpha tubulin and beta tubulin. A large number of additional cellular molecules called microtubule associated proteins, in short we call that MAP, bind to the surface of the microtubule and determine and control its many functions. It is widely accepted that many of these MAPs play crucial roles in the regulation of microtubule polymerization and dynamics, in the organization of microtubule arrays, and in the functional interactions of microtubules with other cellular components. Now the structures, how do they look like? Microtubule polymers are composed of alpha beta tubulin heterodimers arranged in a uniformly repeated head to tail fashion. Each linear array of tubulin heterodimers is called a protofilament, and in cells generally 13 protofilaments arranged into a tube called microtubules. Please follow the diagram properly. However, many examples exist in which microtubules have a different number of protofilaments, from 11 to as many as 15. The 13 protofilaments are aligned in parallel with the same polarity. Beta tubulin subunits of all protofilaments face one end of the microtubule, and the alpha subunits face the opposite end. The ends are designated either plus or minus based upon their different dynamic and structural properties. Beta subunits are exposed at the plus ends, that is called as the fast growing end, while the alpha subunits are exposed at the minus ends, that is the slow growing end. A singlet microtubule made up of 13 protofilaments in cytosol is very common, but many specialized microtubule arrays also exist in which Microtubules are fused along their lengths into doublet or triplet microtubule structure. Doublet microtubules consist of 10 or 11 protofilaments of a second microtubule fused to an entire 13 protofilament first microtubule, and triplet microtubules have an additional 10 or 11 protofilaments of a third microtubule fused to the second microtubule. Doublet microtubules occur in long shafts of motile cell appendages called cilia and flagella. Triplet microtubules occur in the basal body structures that in most animal cells are situated at the base of cilia and flagella, and in centrioles which are situated at the center of animal cell centrosomes. Here is the picture of actin filament. So this is another type of the cytoskeleton, rather the cytoskeletal filaments. What are these actin filaments at all? Actin filaments, also known as the microfilaments, are two-stranded helical polymers of the protein actin. They appear as flexible structures with a diameter of 5 to 9 nanometer, and they are organized into a variety of linear bundles, two-dimensional networks and three-dimensional gels. Although actin filaments are dispersed throughout the cell, they are most highly concentrated in the cortex, just beneath the plasma membrane. Actin filaments, as we know, also called as the microfilaments, is made up of G proteins, or rather the G actins. The globular G actin is a 43 kilodalton ATPase, and like tubulin, it has a binding site for a nucleotide. But the nucleotide is ATP or ADP rather than GTP or GDP. Each G actin contains a magnesium ion complexed with either ATP or ADP. G actin is a highly conserved cytoskeletal protein that is present at high concentrations in nearly all eukaryotic cells. It is the most abundant intracellular protein in most eukaryotic cells. As for tubulin, the G actin subunits assemble head to tail to generate filamentous F actin with a distinct structural polarity. Two parallel F actins twist around each other in a right-handed helix to form an actin filament. The structural polarity of actin filament is created by the regular parallel orientation of all of their subunits. Actin filaments are thinner and more flexible and usually much shorter than microtubules. Like a microtubule, an actin filament is a polar structure with two structurally different ends. One a slow growing minus end and the other a faster growing plus end. The minus end of an actin filament is also referred to as the pointed end, and the plus end called as the barbed end. The plus end polymerizes at up to 10 times the rate of the minus end. So you can understand that the plus end will increase at a faster rate. The role of ATP hydrolysis in actin polymerization is similar to the role of GTP hydrolysis in tubulin polymerization. In neither case is hydrolysis required to form the filament. Instead, it serves to weaken the bonds in the polymer and thereby promotes depolymerization. The in vitro polymerization of G actin occurs in three sequential phases. First phase, termed the nucleation phase, it is a lag phase in which the G actin aggregates into short oligomers. Then is the second phase. Here the nuclei are rapidly elongated by the addition of the G actins to both ends of the oligomers. As the F actin filaments grow, the concentration of soluble G-actin monomers decreases. Then is the third phase. This is the steady state phase. Here the equilibrium is reached between filaments and monomers. The G-actin monomers exchange with subunits at the filament ends, but there is no net change in the total lengths of the filaments. So the spontaneous initiation of actin filament assembly requires the formation of a trimeric nucleus in a process that is called as nucleation. Now look at the structure of the actin filament. You can already find here there is the plus end and the minus end having a girth of about 7 nanometer, and the small black dots indicate the ADP which is bound to the actin filament. So now children, I think you have understood, rather you have got an idea about the different types of the cytoskeletal filaments that are present in our system. Now I have talked about the intermediate filaments, then the microtubules, the actin filaments and so on. Here I am showing you that there are certain drugs that affect the microtubules and the actin filaments. These microtubule and actin filaments are the targets of a variety of drugs. Now these drugs just act by interfering with the polymerization and depolymerization of subunits. Here I have just depicted in a tabular form the different drugs which are acting on the actin filaments as well as the drugs which are targeting the microtubules. You can find that I have discussed about the way these drugs affect the functioning of the actin filament and the microtubules. Now look at this slide. Here I am showing the organization of the cytoskeleton in a polarized epithelial cell. This is a very vital one. This polarized epithelial cells are a very good example of the action of the cytoskeleton. All the components of the cytoskeleton cooperate to produce the characteristic shapes of specialized cells, including the epithelial cells that line the small intestine. At the apical, look at the upper part, apical surface facing the intestinal lumen, bundled actin filaments form microvilli that increase the cell surface area available for absorbing nutrients from the food. Just below the microvilli, a circumferential band of actin filaments contributes to forming cell-cell junctions that prevent the contents of the intestine lumen from leaking into the body. Intermediate filaments are anchored to other kinds of adhesive structures, including desmosomes and hemidesmosomes, that connect the epithelial cells into a sturdy sheet and attach them to the underlying extracellular matrix on the basal side of the cell. The microtubules run vertically from top of the cell to the bottom and provide a global coordinate system that enables the cell to direct newly synthesized components to their proper location. So here I am just wrapping off the discussions regarding the different types of the cytoskeletal filaments. The remaining one, that is the accessory filaments, or rather the accessory proteins, which are a very good helping hand, or rather an indispensable part for the functioning of these cytoskeletal filaments, will be uploaded shortly in the next slide. Thank you.